So the complement system is basically a group of proteins that activate one another to destroy invading microorganisms. So there are circulating polypeptides in the blood produced in the liver that are part of the immune system, okay? And so what it does, again, like I said, it gets rid of or invades or kills foreign substances such as pathogen, microbes, bacteria, okay? And there are three different ways that we can kill the foreign substances. So the first way is opsonization, and basically, if we, let's see, this is our bad guy. Ooh, angry, we do not like him, okay? So the first way is that our complement proteins will come and coat this bacteria or pathogen and makes it taste better for this macrophage, this Pac-Man, okay? So remember, this macrophage already loves the pathogens, okay? That's their job. They love to eat these foreign substances. But when you coat the pathogen with the complement proteins, oh my word, it's heaven on earth. These macrophages devour these guys because it makes it taste better, okay? So essentially, opsonization is the enhancing of phagocytosis by making the pathogens tasty, making it taste better for the macrophages. So making the pathogens tasty for the macrophages, okay? So that's the first way we can kill and get rid of the bad guy, okay? The foreign substances. The second way is the membrane attack complex, MAC, okay? And what it does is basically you have this bad guy again, you know, bad dude. What the complement proteins come and do is basically poke holes in this guy, okay? So it's poke holes in this foreign substances, in this pathogen. And what it does is it causes osmotic imbalance, okay? And that results in cytolysis, breaking of the cell, okay? Because the fluid, once you poke holes in it, the fluid comes in, and the pathogen is filled with this fluid, and it basically swells and pops, it breaks, okay? So that's the second way we can get rid of or kill pathogens. The third way is enhance inflammation, okay? And essentially, what it does is these complement proteins will bind or interact with the lymphocytes. And again, lymphocytes are a type of white blood cells and can help with inflammation, okay? So that's just kind of the summary. So those are the three different ways that the pathogens can die, okay? Those are the three types of deaths they can have. But there are three different pathways in which we can determine how they die, okay? So the three different pathways, they all end in the same thing, which is this complement protein C3. But there are different ways and things how it can activate this specific protein, and that's what we're going to be talking about, the three different pathways, okay? So the first one, let me get a different marker since it's dying. So the first one is the classical, okay? And for this classical pathway, in order to activate the complement protein, we need an antibody attached to an antigen, okay? So that's how we activate the first complement protein, C1, okay? So once we activate C1, it's going to cleave off or split into C2 and C4, okay? Complement proteins. They're still complement proteins, okay? So again, it's a complement proteins activating other complement proteins. So it's a cascade of things, okay? So once we have C2 and C4, they're going to cleave off into their own little thing, proteins. So C2A, C2B, C4A, C4B. And these two guys, they are best friends, I tell you. They love to work with each other. So they work with each other to activate bada boom, bada ching, C3, which is what we talked about up here. Because guess what? This guy determines the 3 different deaths the pathogen can have. So, again, very important. C3, once it activates C3, it's going to cleave off to C3A and C3B. And they're important because C3A helps with inflammation, okay? We put inflam for inflammation. C3B helps with OP, opsonization, and MAC, membrane attack complex, okay? And I'm gonna box this ending results because, like I said, the ending results for each pathway is the same, but the activation process of the complement proteins are gonna be different, okay? So that's the first pathway. The second pathway is going to be the alternative pathway, okay? And so essentially, the alternative pathway is a little different in that it requires these specific complement proteins called B, D, and P factors, okay? So these are specific CP, complement proteins, you should know that. So what they do is they interact with the pathogen and basically activate C3. Bada boom, bada ching. What is C3? It cleaves off to C3A, C3B, and we know inflammation, opsonization, membrane attack complex, okay? So that's the second way, second pathway. The third is going to be, let's see if you can see, lectin pathway. And lectin is actually a complement protein. So in order for us to activate C3, right, we're going to need lectin plus mannose, which is a carb or sugar, interacting with the pathogen. Once they do that, it's going to activate, guess what? C2 and C4. So this lectin pathway is very similar to the classical pathway, because it cleaves off or it activates C2 and C4. And again, C2 and C4 cleave off to their own little proteins, right? So C2A, C2B, C4A, C4B. And remember, these two dudes right here, C2A and C4B, are best buddies. They are the best partners. They love to work with each other. And they activate C3. And again, cleave off to C3A, C3B. So, if we step back and look, as you can see, sorry, I don't have the best camera quality. So if you step back and look, the way that they're activated, that this protein is activated, C3, is different. The classical requires the antibody to attach with the antigen. Alternative requires these specific factors to interact with the pathogen. The lectin requires lectin and mannose, which is a carb or sugar, to interact with the pathogen, but they all produce C3, right? Which results in the same thing, okay? So how they're activated is different, okay? Up here, it's gonna be different. But then eventually they dwindle down to being the same thing, producing C3. And again, what does C3 do? What it does is that it determines the 3 types of deaths the pathogens can have, okay? Because these are the 3 types of deaths. opsonization, MAC, membrane attack complex, and the enhance of inflammation. Hope that helps.